colony formation is inhibited pretty well.
Good. We can test the BC-PAP cells. Cell lines are really the workhorse of biomedical research. Having the right cell line models is critical.
More than 95% of the traditional, if not 99% of the traditional classical cell lines do not have an original sample to which to compare. A lot of labs are still ignorant of the degree of cross-contamination and mix-up of cell lines. They're ignorant how big of a problem it is. 2 out of 9 cell lines are misidentified. Can be due to mix-ups, can be due to mislabeling. 2 cell lines that have the similar names.
When I started working on thyroid cancer in 2006, I collaborated with an internationally recognized expert in the thyroid cancer field to obtain thyroid cancer cell lines. This collaborator had received these cell lines directly from the originator. About 6 months later, I was really excited about some of the results I had. It was just perfect result that you would dream about. It really was too perfect. That's when I started to doubt the origins of the cell lines.
What Rebecca noted was that there were a lot of things that were irreproducible, different behavior than they expected from the cell lines. We started to accumulate cell lines and test them for what their identity was.
We found that 19 out of 40 of these cell lines that we obtained from around the world were misidentified or redundant with other thyroid cancer cell lines. It was shocking. These thyroid cancer cell lines had been used for the past 20 years. My lab is interested in identifying new therapeutic targets for thyroid cancer. We like to authenticate cell lines in our laboratory on a regular basis. We have a verified liquid nitrogen tank. Every stock of cell line that goes into that tank has been profiled by STR genotyping, and they've also been tested for mycoplasma.
We use sequences in the DNA called short tandem repeats, or STRs. That's not always been done.
There are big financial costs for not authenticating your cell lines. Some of the latest data is showing at least 30,000 publications, including citations, are using misidentified cell lines. I think the latest estimate in wasted dollars is around $50 billion.
On average, each of those publications over its lifetime gets cited about 15 times. So you start building up a pool of this upside-down pyramid that's building on other pyramids, and as soon as you start pulling one pin out, the whole scientific literature collapses.
As one of the leading providers of biological materials, it's really our responsibility to be a leader in the authentication field. We employ the stringent QC and authentication whenever we go to produce. All our material in the ATCC collection is traceable. You buy it from ATCC and you're assured of what it is.
In the regulated environment, there's no choice of doing authentication. It's required. When you file your applications, the agencies will look to see if you followed those regulations, those guidance documents. And if you haven't done it, you won't be successful, period.
Drug developers, you have to make sure that the diagnostic has to be to the right material. If you don't have the right material as the control or as the source of developing the diagnostic, then that's really going to have a significant negative impact on the quality and the effectiveness as it's released to the public.
Why waste your time with cell lines that are not what you think they are? Scientists are very collaborative. We don't like to say no. When a colleague requests a cell line, I think it's good to say no because it's ethical, not to share cell lines that may or may not be authenticated.
We see it as this is an important time to establish a single source of truth. Doing the authentication using not only our previous methods, but then adding in the whole genome sequencing. And this becomes composite data. It's establishing a baseline.
Authentication is becoming more prevalent in biomedical research. It is getting on people's radar.
I think that this indicates a changing tide. getting this into the practices of the younger generation. So as they move forward in their scientific careers, they're bringing the authentication practices with them.
Yeah, so this data looks really good. You have nice, even peaks. I see the future of authentication really gaining momentum. This will be our first generation of scientists that recognize the problem and they will know how to deal with it. 100% match, you can't get better than that.